Hello, good evening everyone. Welcome to the first open mic of 2025. It's quite impressive to see how far we've come in the last year since we launched the event series on the Novartis Pavilion in May of 2022. This is actually already open mic number 13. And I think it's fair to say that we can already start calling it an institution on the campus. We have another interesting program lined up for tonight. We're going to hear a discussion about innovation in precision medicine and biomarker development. I'm really looking forward to an insightful discussion. My name is Rahel Schneider. I am hosting this event series with Switzerland Innovation Park Basel area. For those of you who don't know us yet, we are operating a small co-working and office space here on the Novartis campus for health tech ventures. The event series is curated by Day One Healthcare Innovation. That's the health tech initiative by Basel Area Business Innovation. And before I hand over to Ursula, I want also to introduce or to welcome everyone joining us online. As always, we are using Mentimeter to fill the Q&A section. So please get involved. Also, everyone on site, you can scan the QR codes on the screen and send in your questions. And Quiva, our moderator, will take them up during the event. But now over to you, Ursula, director at Day One.
Thank you so much, Rahel, and thank you everyone for joining us here and for joining us online. I think after Rahel's intro, there is very little more to say, and I know most of you that come regularly got tired of seeing my face up here. So today I have a big surprise for you because it's a real pleasure to introduce Quiva Vallely, so Director of Strategy and Pharma at Day One, as the moderator for the event this evening. And besides that, I just really wish you insightful but fun and full of insights evening, and that you all get to enjoy. So Quiva, floor is yours.
All right, thanks very much, Ursula, for the introduction, and I'll extend my welcome to everybody as well. Some of you may know me already or have seen me around, but this is actually the first time I've actually been on stage for an open mic for Day One, even though I have worked for Day One for over 2 years now. So essentially what we're going to do today is we're maybe going to try a little bit, not differently, but try and give a little bit of an interactive approach to the conversation that we're having today. We should have been 3. I know that you're aware that there were 3 of us announced for the panel, but very unfortunately, Noé has been struck down with flu. So we wish him the very best. And we hope that he gets better and is able to join us at another stage. So what we've done is we've pivoted very slightly. While we're still going to focus on innovation in precision medicine and biomarker discovery, what we're going to do is look at it from 2 different points of view. From the point of view of a very established company who is leading the large-scale usage and implementation, but also from a small novel tech company and what they're doing differently within precision medicine and biomarker discovery. So as I mentioned, or as Ursula mentioned, my name is Quiva. I've been with Day One and Director of Strategy and Pharma for 2 years. Previous to that, I spent about 18 years in the pharmaceutical industry, of which 15 were in precision medicine and biomarker development. So this is why I'm stepping in to moderate on this one. So I'm going to have an awful lot of opinions during this as well. I don't really sit back and do too much not speaking. But on top of that, just to let everybody know, I have Mentimeter. Ursula's going to be in charge of sending me the text messages for Mentimeter. So I promise you, if I'm looking at my phone, I'm not on Instagram. I am actually trying to work your questions in live to the session. So what we're trying to do is if you've got a question on a particular thing that we're actually talking about at the time, we'll try and pull those questions into the conversation so that they don't get lost or diluted or out of context later on. So I'm going to stop there and I'm going to introduce you to our 2 panelists. As I mentioned to them earlier on, I absolutely hate introducing panelists while they're sitting there looking awkwardly. So what I actually will ask our panelists to do is introduce themselves. So I'm going to start with Sarah, who comes to us from Scale Out. So Sarah, if you want to go ahead.
Sure. Thanks, Quiva. My name's Sarah Carl. I'm Director of Data Science at Skylight. I think I'll have a chance to talk some more about that later. But in terms of background, I have a PhD in genetics. I've worked as a bioinformatician and computational biologist at the FMI here in Basel for several years, which is affiliated with Novartis. And then I joined Skylight in 2018. So the company was founded in 2017. I came in fairly early on. And yeah, I again don't want to talk too much about the exact role now, but that's sort of my background in mainly analyzing large molecular data sets with both classical sort of computational techniques and now more machine learning AI.
Great. And then we have Elke from Roche Diagnostics. So
Yeah, welcome everybody. Yeah, it's a pleasure to be here. I'm actually in Roche Diagnostics since about 4 years, working at a, let's say, novel unit that works into the early innovation space. I call it the experimental think tank that we built up 3-4 years ago. And before that, I've been 4 years in pharma, also working already on the early discovery side of things, and also led an academic team in the past. So pretty much on the innovation, you can say, all over. And right now, our shop is really looking into a lot into PHCS or precision medicine and biomarkers and novel biomarkers and platforms, what we can do for the patients. Yeah.
great. So I think, you know, if we start, maybe let's do first of all a kind of an overview or a scene setting of what precision medicine is, because having worked in it for quite a long time, I know that everybody tends to have a very slightly different understanding or definition. So if we all kind of give a quick definition to you, Sarah, what does precision medicine mean to you?
Well, at the end of the day, I think precision medicine is about finding the right treatment for the right patient every time. And it sounds simple, but obviously it isn't. I think that's the end goal, and then you can take many different steps along the way to get there. But I think precision medicine is about breaking down what may have been looked at in the past as a monolith, like an entire patient population, breaking it down into individual patients. But even within one person has many factors that can possibly contribute to how they will respond to a certain treatment, from environmental factors, molecular genetics, lifestyle, etc. So it's really trying to go down to a much more granular level at how we understand diseases and treatments.
Elke, anything to add to that?
Yeah, no, I mean, that's completely right. I think it's about the patient and the individualized healthcare treatment. We come already to an opinion. So I do think precision medicine, in a sense, I mean, that's something we are aiming for. I would be already happy if we go towards more personalized medicine, which, at least from my perspective, is already a step forward in terms of looking up multiple more novel biomarkers, et cetera, et cetera. I think then we would already be doing pretty well.
Yeah. So great. And I think I'll maybe add on top of that, because Sarah, you mentioned that it's the right medicine to the right patient, which blurs a little bit with the personalized medicine as well. But I think where precision medicine can go one step further, that I don't think we talk an awful lot about as well, is personalized dosing as well. It's not just the flat dosing structure of a particular medicine, but it is what is the right dosing level for the individual patient with the specific demographics. So I think that's something else that biomarkers specifically can be very, very well leveraged to get to that level of dosing. So I think from that point of view, if we flip into, we kind of now have a very comprehensive view of how we feel precision medicine, personalized medicine is used. What is it that you think that your role specifically, if we start with Elke on this one, what is it that you think your role does to contribute to personalized and precision medicine?
So at the moment, we really launched like 7-8 new projects that is somewhat adjacent to the Roche portfolio, but somewhat also new. And I do think it makes a step forward really on the later, we talk 5 to 10 years from now, innovation. So anything in terms of earlier diagnosis, so most of our projects, if we look at biomarkers, are into the earlier diagnosis. We call it the early symptomatic people or pre-symptomatic. And so those studies are very complicated. So you can imagine what kind of studies would you conduct, which are the controls, etc. And so the study size is even much bigger. So the invest of the company is even potentially much bigger. So, and you can probably talk about that with the data that you are getting from that. So earlier diagnosis is probably one of our major themes in our platform. On our shop that we look at, or it's platforms that are not existing yet but enable a novel diagnosis. So for example, we look into platforms for aggregation of proteins, which currently we can't do so well in Roche, so we just measure actual standards of proteins right now. Or of course we also have genomics and all these other platforms. And that's not so easy, so that would be a novel platform in case we manage. And that would enable actually the diagnosis of Parkinson and other diseases as it currently looks like.
And Sarah, for you, what on your day-to-day role is really targeted towards precision and personalised medicine?
Well, really the mission of Skylight is to advance precision medicine by combining the technologies of single-cellomics and AI. So my role as Director of Data Science is leading our data science team, which does both algorithms development and data analysis. So we're trying to bring these technologies to bear. We work with different data sets that are coming from either clinical partners, university hospitals here in Switzerland, also pharma companies, biotechs that we've partnered with in the past. And we try to do biomarker discovery, in particular, going to a new level of sensitivity and resolution with the single cell data that would enable us to attack difficult questions. So I think it's a different angle of difficulty than early diagnosis in really large patient cohorts. I think that's great to hear that Roche Diagnostics is working on that, because you really need the resources to scale that a large pharma company can bring to that. So we have tended to focus more. We have actually had some project in diagnostics, for example, in endometriosis, which we recently sold a biomarker in that field. But now we're focusing, I would say, more on patient stratification for treatment. And in particular, we've recently been awarded a grant from InnoSwiss to work on generating a large cohort of data from IBD patients from a Swiss biobank, and look for biomarkers to predict treatment to different biologics in that disease area.
Great. So moving on from that, and I know Ursula's done a great job, so folks, I am aware questions are coming through, and if I'm not addressing them immediately, it's because I know I have similar ones already prepped and ready to go. So I will loop them into the storyline, I promise. If we move on from that, and one of the questions came in is very similar to this, and essentially, so like I mentioned, I've worked in the clinical space for almost 20 years. And from the day and hour I went into the clinical space, it was very much biomarkers are quite standard. This is not new. Obviously, I worked in a biomarker department, but it was not new that they were incorporated into clinical trials. But they were pretty standard. They were not super inventive or anything like that. They were just, this is what you did. They were well accepted. You used them. You moved on. But what I've seen outside of that, and if we're looking at the clinical space versus what we are actually understanding from a technological advancement and a disease advancement, is that I still think the implementation is lagging behind our technology and our science. So I think that the ability for that discovery has accelerated exponentially, but we're still not quite really getting the use out of this. So Elke, from your perspective within the industry and looking at how things have moved, what do you think has been the biggest, what has impacted the use and the discovery of new biomarkers the most in the industry?
I would say, at least I can notice there's a mindset shift. At least I can tell for sure within Roche, particularly since the CEO is leading us now. There's a mindset shift which enables us more to collaborate even within the big company. I mean, as you know, we are huge. So actually within the company, I feel like it's essential that pharma and diagnostic works together, which in part worked, but not as such that, for example, and we talked about this before, is that pharma is open for better patient stratification and really enabling new biomarkers. And I do think we are moving into a space within the company. One is the mindset. The other is also the external pressure. So we know that the healthcare system is going through a transformation itself in the country. So we have global health burdens. We have to reduce the costs. And probably medicine is not going to be as expensive anymore in the future. And health industry, like the companies will not reimburse it, or the health insurances. So there's a lot of pressure also from external to make better patient stratification also happening. So I do think it's coming also from external pressure in terms of prices, to be more clear on which patient do my medicine work, so that I also reduce the costs maybe in the end, even for production, etc. But I'm very clear, these patients will benefit from this kind of drug. So I do think it's a combination of both. And I can tell for sure with them that the part that I'm leading, that this has really enabled a lot the last 2 or 3 years, that we have many projects internally from the beginning on design together between pharma and Dia, which would make a huge difference in the long run.
Yeah, so essentially we're moving towards we can't afford not to implement these more structured strategies, as opposed to when we were delivering on clinical trials, novel biomarkers were nice to have, but they were not necessarily had to have. So they weren't primary or secondary endpoints in the trials. They maybe floated around in the exploratory endpoint side. But with the cost of generally the world increasing and the access to medicines cost not being able to afford, and frankly, if we're being very honest, the research and development budgets in pharma are not exponentially growing either. So we can't afford not to be leveraging this technology and this knowledge as a must-have.
Yep.
So we talked a little bit about, is technology outpacing our ability to use it as yet? So if we switch from thinking about how we're trying to implement in large, quite established companies, and we move towards, thinking about Sarah and what you do in the cutting edge space, You work for a company that is working on the development of novel biomarkers using that cutting edge technology. Can you give us a quick understanding of your product and tell us what are the advantages of using single-cell omics for biomarker discovery?
Sure. So our product is essentially an analytics platform. So we're not building new hardware or new assays. We use existing hardware and assays that are out there. We work a lot with 10x genomics, single-cell assays, mainly RNA and also multimodal RNA and protein. But where we bring something novel is the way that we analyze the data. And this is very complex data. It's very large. It can take, in an academic setting, a PhD student or a postdoc could work for months or years on these data sets. Our approach is very targeted, so we use something that's called supervised machine learning, and that means that we have an endpoint in mind that we want to be able to predict. Let's say it's response to a treatment, and we're going to train our AI models to predict that endpoint, and specifically by finding the patterns in this data set, this large complex data set, that are most directly relevant for that endpoint. So we're not trying to characterize every tiny, minute detail, which an academic project might go into. We really want to see, is there something there or not that's going to let me stratify these patients? And if there is, then we can dig into it and we can go in more detail and we can start to investigate the biology, but also try to build simplified models that could then be translated. And I think we'll talk about that maybe later. But that's an overview of our approach and our product. And I think there's a second part of your question that I've forgotten now.
Yes. Nope, that was pretty much it. So it's understanding the product. Oh, yes, sorry. And what are the advantages of using single-cell omics?
Yeah, the advantages. Well, really, the advantage is the granularity and the resolution that you gain. Mm-hmm. One metaphor that's been used a lot, but I think it's still apt, is thinking about kind of a fruit salad versus a smoothie. So if you're trying to find out, what are all the different components, and if you want to find, you have a fruit salad with one type of fruit in it that's gone bad, if you dump it all in a blender and make a smoothie out of it, that's kind of the bulk omics approach. And then you have to figure out from there, okay, what went into this? How can I tease this back apart into its components? And for a long time, that was the technology we had. We could do a lot with that. But now we can do better because we can directly look one piece at a time, one cell at a time. And so to take it back to the real world, let's say we're looking at an immune system, we're looking at immune cells from a blood sample. Instead of mixing them all up together and getting an average profile across all of the different types of immune cells, we can really say, here are these specific types of T cells, here are these specific types of B cells, here are these specific types of monocytes. Which of these are contributing to the signal that I want to understand in the end, and which of them are just bystanders that don't have any effect there? So I think that's really the advantage you get from single cell techniques.
Elke, do you have much experience in that as well?
Actually, yeah, we have a little exploratory single cell project actually in the US that looks into where we diversify our tissue with a novel technology of the surrounding tumor. And big collaborations actually with, and I guess we come to that too, and big collaborations also with Europe, from US to Europe and then back to Mayo. So we are part of Manifest, for example, with that technology, which is this big UK-driven but European-centered consortium looking at to better prognosis of cancer. And that technology is actually from our shop a novel technology that we have proprietary IP on, and that is really early innovation space where we started with blenders. I'm not smoothie. We started with blenders, but we generate single cells with a formalin fixed from formalin fixed tissue, and then we sort ourselves even, and that is now being integrated into that consortium. So we truly think in single cell as well, and obviously people probably know Aviv Regev being part of Roche, driving a lot on the US side on single cell for pharma as well in reverse translation. So I do think the field is moving towards that space for sure.
I'm just going to stick on the technology side of things at the moment, and I'm going for our first question because we've hit at the right time. So the question I have on the Mentimeter is technology to measure aggregation of proteins. Could you please explain a bit more and give practical clinical examples where this adds high medical value? Who would like
I
to
guess
start?
that was to my reference of the aggregation of the proteins. Yes, happy to do so. So in Parkinson's disease, actually currently people use alpha-syn as the biomarker. Alpha-syn is a protein that sits in the brain, and through the progression of the disease, it starts to aggregate. So you can imagine, probably all of you know Creutzfeldt-Jakob. So in many diseases of the brain, it's right now thought that these are prion-like proteins. In some diseases like Parkinson, it has been more clear now that the aggregation of alpha-syn is more the indicator of the disease than alpha-syn itself or already aggregated alpha-syn. So what one would need is actually a platform that can read out the aggregation over time as an indicator for Parkinson's disease. And that is not easy to detect. There's a company that has, I think it's also a startup, they have a clear lab certified test out, as far as I know. And of course, Roche intent is to do a standardized one-day IVD platform if we manage. And so at the moment, our idea is to have a quantitative, qualitative, robust assay in place. And we get started with buffer, and then it gets more complicated. Then you can imagine CSF, and of course the holy grail would even be blood, whether that's even possible biologically to be seen, and then also technically. But yeah, so that's a background, and so we believe this could be a platform for multiple neuro diseases in the future. For p-tau, this has also been reported. Right now, p-tau 217 is the marker currently believed in the field. We also have this on our platform. That's just a current novel or protein marker that got, I think, breakthrough achievement last year. But that's a current standard, but there's a lot of literature already indicating that also for Parkinson, the aggregation of p-tau might be essential over time.
Great. So you mentioned that there was a specific startup that was developing those assays. So I'm going to flip back to Sarah here for a second, because one of the very important things that we see is that technology and those assays and the biomarkers are coming, a lot of the time they're coming from quite small companies, and they're very, very focused. So from your perspective, you develop your novel biomarkers, but how are you planning to scale these discoveries so that they become routine clinical practice and standard of care for patients?
Yeah. So in our case, there's kind of the immediate path and then the future path that we could envision. So right now, essentially our approach is to scale them by translating them to currently existing clinical technologies. And that depends on which type of molecule we're measuring. So if we're working with cell surface proteins, that could mean translating a biomarker that's first discovered in a large, unbiased assay into a flow cytometry panel, something that can be run on a CE-IVD instrument. If we're talking about RNA, it can mean translating into a panel that can be measured in some bulk assay like a qPCR, or potentially a sequencing platform. Roche has some interesting developments in that space that's come out recently that I think will be changing things as well. And that kind of leads towards what I hinted at with the future, which is I think that as newer technologies, particularly in the omics space, are being developed and being commercialized, I do envision that the price of those technologies will come down. Also in the single cell space, there are a lot of small players now that are starting to bring new technologies, but also I mentioned 10x Genomics earlier. They have a new kit out that works on FFPE tissues as well. So that really facilitates applying things to both retrospective samples and collecting samples in ongoing clinical trials. So basically, I think the future will probably get higher dimensional and enable these kinds of new technologies to be deployed at scale, but I think it will still be a while until we get there.
Okay. So I've had a question come in for you, Elke, and I think maybe it requires just a little bit of clarification. So the question I've had is, alpha-syn is however not specific to PD. How do you deal with that? But just from my side, is alpha-syn, is it the aggregation that is specific to Parkinson's outside of other conditions, or is the aggregation reflective of other neurological conditions as well?
Well, alpha-syn itself, yes, it can be a biomarker for other diseases as well. But yes, it's the aggregation over time that's currently, to my knowledge, the standard of care, and also that pharma is looking into to put their clinical trials according to that. Yeah.
Okay. So great. Sorry, I'm just moving questions around. Brilliant. And then we had another question that I think I'm going to loop into it as well. So Sarah, how easy or hard is it for you to get the clinical data for training these modules that you have?
Mm-hmm. It's not that easy, but it's getting easier. I'll put it that way. So first, on one side, there's more and more data that's being published. So that's one source that we can go to is looking for public data. But also the companies that we've been approaching and working with in the past, I see that they start to proactively want to incorporate these technologies into their clinical trial designs. And as you said before, it's mostly still on like exploratory arms, this kind of things. It's not a primary endpoint, but we start to have more companies, especially in the sort of mid-sized biotech space where maybe they can be a little bit more agile, that say, yeah, I want to collect single cell data in this clinical trial. Can you help me analyze it? So I guess that's one aspect of the question. The other aspect would be coming at it from, how difficult is it to generate that data in terms of a wet lab flow? And I think that's also gotten a lot easier and more standardized in the last 5 years or so. So I think there were, when we first started in this space, it was a lot more challenging to have really reproducible, robust data, and I think that's improved a lot.
Okay, great. I'm going to move back to you for this one, Elke, because I've got a number of questions that have come up relevant to this. So obviously Roche Diagnostics is one of the biggest players in the established biomarker platforms and technology space. How do you work with companies like Sarah's to collaborate with with academia and startups to really, or on tech transfer to the larger platforms. So what's your kind of approach to that?
Yeah, so I can definitely talk about this because that is one of our mandates. So we are called Discovery and Open Innovation, even on the diagnostic side. So on our case, on the biomarkers, I mostly refer to, we mostly work with academics and big KOLs actually, because they have a lot of the biology and really the expertise to dive into the disease biology that we are currently interested in. I mean, let's put it like this. We do collaborate and we get many contracts signed. It's also not easy though. So to the point we have reached an agreement how this could look like and who owns what and how we maneuver through the time, that might take a year or two till we manage that agreement. But we do manage. We have on the German side quite a few big collaborations. We've just worked on we call it the Pandemic Alliance Munich, so which is many universities working with us together. Also on the US side, we just signed a deal with Mayo on the early biomarker space to work with diagnostics and Mayo and their clinical trial samples and some of their teams. So we did manage those. It did cost us quite some time. With startups, it's a little bit different. So either they are extremely small, and then usually we then might even go into a collaboration agreement for a while, where usually we just try to see a bit the space, and the startups profit from it because they get some advice and they see a bit, they get networks, etc. Once they get a bit bigger, it gets more into the space of probably, do we want to acquire them rather than collaborate? So my experience is we either collaborate with really small ones or we watch the space and acquire maybe some. I mean, that's my experience, at least. Yeah.
And that's actually where the link to one of the other questions that we have. So one of the first questions that I got was, what capabilities do you prefer to build in-house and for which would you partner? Do you have a preference on that at the moment?
For which do you what?
Capabilities do you build in-house?
Yes.
Or partner?
I did already answer it a bit. So in my view, from the biology expertise for biomarkers, I tend to partner. So I've run myself an academic lab just 10 years ago, also looking into the immunology space. And to me, it makes sense to really partner with academics. Usually they're quite happy to collaborate, to get industry knowledge. They have the deep expertise in the disease biology, which to me takes a long time to build it up. And then for technologies, as I said, it is, if they're super small, yes, potentially also a partnership over time. But then we usually rather watch and observe and might actually acquire rather at some point. Yeah.
So I'm going to move on a little bit to kind of a twofold. So we've talked about where we are at the moment, where we're established. You touched on it very briefly, Elke, when we talked about the actual biological measurement space, whether it is CSF or spinal fluid within the Parkinson's, or whether we're able to actually progress and do a ratio to blood. So we see a lot of our biomarkers, the most accepted biomarkers, tend to come in relatively standard biological tissues. It can be quite invasive, such as the spinal fluid or tumor blocks that are coming from patients. And is this enough, or do we need to be pushing into more novel biomarker collection spaces? Unfortunately, Noah, who was going to come, he actually works on biomarker development in sweat, which would have been a really interesting segue in this conversation. But how often do you see, when you're having the conversations with innovative development, how often do you see the biomarker measurements in novel materials? Or do you see mostly in standard biological material?
Well, for us, since we are big and I always call us the Titanic, which is difficult to move. You know, you see it. So anyway, so that's why I'm thinking.
It also sinks. Maybe.
No, don't say this.
Exactly. No. That's why I'm like, I come from Belfast. You know, this is.
Don't say this. I just meant the metaphorical, you know, movie. So yeah, that being said, so I still think it's a classical one. From a business perspective, this also makes a lot of sense. So if you look at the moment how we in diagnostics thrive, it's through the platforms. So the platforms offer a certain specimen in most cases. So if you look at Core Lab, the IVD current bigger instruments, they are automated for usually plasma in most cases. Some biomarkers are measured in different, maybe whole blood or so, but overall you have mostly a automated plasma separation, then it moves further. So all of this, just to give an example, makes sense. We know this from tumor. I mean, we have our platform centered around it. So we usually as a company have quite a difficult time to accept the early innovation space, even for, so it correlates with new technologies and new different specimens. But we are moving. I mean, we have a lot of, particularly in the research space on cell-free DNA, obviously for cancer, other type of detections, because the field has moved quite a lot. Roche internally, as you might know, has also, for example, ophthalmology, quite a lot of pharma drugs. So people are interested in teardrops and biomarkers on teardrops from the pharma side, and we haven't set this up in diagnostic yet. So there is, I think, interest to move into that space. But one is a technology burden for a big company, so moving into a new platform, the usual topic. And then the second, I mean, I'm myself a biologist, so it might even not be possible. So it's a combination of technology, but it might also just not be true. You find a biomarker, let's say in blood, of a neuro diseases. It might just not work. I mean, even if there's a novel breakthrough of technology. I mean, technologies enable a lot and enable breakthroughs in biology, but it might also just not be there. So I think it's all about research and trying it out. Yeah.
Sarah, have you any thoughts? Are you working primarily in standard tissue, or are you looking at anything alternatively?
So I think Elke's last point was really important for us, especially. Yeah, we follow a lot the biology, so it has to make sense from a biological standpoint. Of course, the less invasive, the better, but that's not always possible. Also, you know, there's a bit of a special aspect. Since we work with single cell data, we need to be able to access single cells. So saliva, for example, is a really interesting potential source of biomarkers, but it wouldn't really work for our platform. Tissue can work if it can be dissociated, which most tissues these days can be. But of course, blood is kind of the ideal source for us. And another one that I think is kind of interesting that we've done a couple of projects related to are actually cell therapies, where you might want to try and find biomarkers in the product itself, which is made of cells, right, that have already been extracted from a patient. So yes, that's an invasive process, but they were anyway going to go through that process. And then we've tried to find biomarkers predictive of response, but also adverse events or manufacturing success, for example, from the product itself.
So if we think about and what Day One does, obviously Day One works very strongly in health tech, and one of the things that stand out for us and we get a lot of discussions around is our friend the digital biomarker. So if we're looking and we look at something as unburdensome to a patient as possible, and we think about the digital biomarkers, the question I have here is, what do you think of digital biomarkers? Are they as useful as biological biomarkers in precision medicine? And if not, why? Anybody got an opinion on that? I know I have. So
super curious to hear.
The more data person.
Well, I can try and start, but I don't know if I'm really have that much insight because I'm not directly working with digital biomarkers, I would say. I think from my perspective, I think they could be useful, again, depending on the context and what it is you're trying to actually find and monitor. If I would think about trying to integrate that into a discovery that my company is doing, so you know, we can I've been really emphasizing that we work with single cell data, but we can also integrate other data types, standard clinical measurements, but also why not digital biomarkers? So we could potentially build a more complex model that integrates these different modalities. Then that gets a little bit more difficult to think about implementing in practice. But that could be one way to kind of combine these different kinds of readouts and capture different aspects of a patient. But yeah, curious to hear from either of you.
So from my perspective, digital biomarkers are fantastic, but they're not standalone. I don't think they provide enough context. I also don't think they're established well enough and reproducible well enough at the moment to really be a standalone measurement. I think that, and having worked in clinical trials where we implemented measurables and wearables, there's an awful lot more user error when it comes to digital biomarkers than there is with the standard biomarkers. I have a large amount of hope that long term they will be very well integrated into people's normal lives so that they become an excellent longitudinal measurement and a prognostic kind of tool that will be really good. But I don't think we're at the stage where they will be standalone either from a diagnostic perspective or from a long-term disease monitoring perspective. So that's my personal opinion on digital biomarkers. And I'm a wearables nerd, so I've spent a lot of time testing every wearable on the market and some that aren't on the market to see, you know, what kind of data that I can get out of my own information.
Yeah, I share a similar opinion. I mean, Roche is indeed investing. Actually, we have a separate unit even built out quite a few years ago that looks into, we call it information solutions. And we look quite a lot into that. We also have this Navify, which you might have heard of, and we are looking further into that. For certain diseases, it makes, of course, neuro diseases, we talked about this, a lot of sense to me. Whether, again, this is a standalone, I'm also not so sure, but to me that would make extremely a lot of sense. We were just thinking of whether we should look into chronic fatigue syndrome. I thought like a combination marker too, where people have apparently a lot of exhaustive measures. You can't diagnose it well as a maybe new project, which I could see also digital markers could work. And there might, but neuro is the obvious to me where this could make a lot of sense. And I also think they should be integrated. To me, they are also comparable, similar. We have a topic also now on inflammation markers. Myself, having an immunology background, I'm also quite certain that there will be not many single markers that are specific for one disease, but in combination with multiple other markers, they are molecular markers, but they, to me, appear similar as an additional, you know, add-on to really see how severe a certain disease is, or whether a medication worked, or et cetera, and already starting to see positive. And the digital ones would, to me, be in a similar potential overall bucket.
And so I've got a follow-on question, and this is, I think, Sarah, relevant to you. Or basically, we had the discussion about that. And essentially, the question that here is, each person who is a patient has quite some dark data it comes to in their data set. How are you dealing with gaps in patient data when looking for personalized medicine? So I think this really is
missing data.
Yeah,
from,
correct.
Basically, yeah, this is a really tricky one, I think. And so, yeah, I think there's kind of two ways that you can deal with it in general. One is being very strict and kind of excluding measurements where you have too much missing data in a certain context. And sometimes we take that approach because that keeps the data set clean, and then you really know that what you're getting out is meaningful. The other one is trying to impute missing values, which I'm a bit uncomfortable with, even though there can be, you know, fairly advanced statistical techniques for dealing with that. I think, yeah, it kind of, I would say it's kind of a case-by-case thing, for me at least, in my experience.
Okay, so if we move on, I've got a lot of questions when it comes to regulatory, folks. People seem to be quite interested in regulatory and AI, so we will get to that. One of the bigger questions that I have early on is, and I've saved it to around about here because we've got into the conversation for a while. How do you see personal biomarkers working when we can't even deliver access to treatment for classical therapeutics? Can we really move the 5 steps ahead from a personal biomarker perspective?
Well, you heard already my intro at the beginning, so I also think, I mean, it's always important to have a long-term vision, right? And I think the healthcare industry needs to have that, and we do have certain tools already there. We look at sequencing, we look at single-cells. I mean, the world is turning, and there's way more possible than there used to be. So we need to keep that pressure high. But yes, I'm with that if I look into reality and pragmatically what's being used, I mean, it's already a big step that we currently integrate immune markers into some measurements in reverse translation, which is like at the moment a step forward. So, but this is not to be underestimated, because if I look at the previous example of what I mentioned, P217, that was also, we had multiple markers on platforms for externals to measure, which is classical just protein markers. How it then works is that the world exchanges on the data on the platforms to measure, and this is how the biomarker got established further and further. And although this is just a protein biomarker, it makes a huge difference for many patients. So I wouldn't underestimate that any step forward makes a difference for potentially X amount of patients. So I would think it doesn't matter. I mean, I'm driving a super early innovation team, so I get frustrated in between. It's like thinking, come on guys, let's move. But in reality, it makes already a step forward if we even on the classical biomarkers make a step forward to personalized healthcare. It makes for a lot of patients a difference.
So what I really liked or found quite interesting in that question, and maybe I want to kind of pop it back so if the person who wrote it can write a follow-up after this, if that's of interest, is that there was the discussion around the access to the classical therapeutics. And I think what we are looking at here, or what we have looked at so far in the conversation, is the development of novel biomarkers and potential new treatments. But what, if any, have we seen with regards to progress of going back to existing therapeutics? So yes, there may be an access issue to getting hold of therapeutics. That's one thing. But if we're looking at, are we ensuring that the therapeutics that are on the market are up to date with the right biomarker stratification strategies, and are the right patients being tested and put on the right therapeutics? I don't see an awful lot of that backwards happening. As we progress with discovery and development, it's all future focused and very rarely going backwards. Sarah, have you any thoughts?
Well, I can just give an anecdote as to why I see this as a challenging thing. And this is, again, from the perspective more of an outsider to Big Pharma, right? So take it with a grain of salt. But, you know, a friend and previous colleague of mine who actually works for Novartis, I saw him give a presentation of some really nice work that he and his group had done on biomarker discovery and patient stratification for a drug that was already approved for some time. And I asked him at the end, so what are you going to do next? This looks great. Are you going to try and, you know, put this into practice? And he said, well, no, because this drug is going off patent soon. So there's no financial interest.
Yeah. Well, I can't speak. We do have reverse translation groups in Roche, which are located under both pharma sides, GRED and PRED. So since I'm not in those groups, I sit in diagnostics, but I work with them a bit. I do see some insights, of course. At least to me, there seems quite a lot of shift. I've been in 2 strategy meetings as a DIA representative last year. It looks like, we look into existing medicines and how to use them for different type of patients, so looking at other patient population. And better patient stratification is for sure a major strategic theme. That probably, I told you the Titanic thing, it might take a while to really see that into practice or being on the market, let's say. But let's say from the mindset shift and leaders looking into that and what we can do, I do see already a shift right now. Yeah.
So let's actually take some time to talk about that and talk about what the business aspect of biomarkers is for large companies. So and I'm going to give my opinions on this as well. So I think that's, I've put my phone down. So that's how you know I'm getting serious on this one. So do we look at the challenges of biomarkers, especially if, and I thought it's really interesting that your friend said, no, it's going off patent, so we're not investing in it. But are we really maximizing? I mean, let's be very honest, pharma companies they're businesses. Whilst they do good things and they deliver good medicines for patients and it's great for humanity, they are businesses, they need to make money. So they need to maximize the value of those drugs, which benefits patients because it means you're looking at larger patient populations to see if there are more patients, different indications that are actually going to be beneficial to use these drugs. But having internal conversations, knowing how much it costs to kind of do the investment into that, having those internal conversations can sometimes be quite challenging. Because then you're starting to, you talk with the business teams and you're saying, yes, but it's really worthwhile investing into these biomarkers and it's really worthwhile investing into companion diagnostics. Yes, it might technically reduce your patient size, but it's going to improve your efficacy. You're going to be superior on the market. And from your perspective, Elke, do you see a lot of the mind shift, the change into looking at, from a business side of things, biomarkers have traditionally been seen as we're going to shrink your patient population, and that's not good because we want to sell as many drugs as possible, to we are going to give you the gold standard patient population, which means that your efficacy is best on the market, best in class.
Well, I haven't interacted with the business side on the pharma side. I can tell you the business side on the diagnostic side, even on the PHCS, so pharma business side, so that directs to the pharma side. Yes, it's difficult. And to be honest, that's their job. So the question is, I mean, they're there to sustain our industry and make sales. So it is difficult to have these conversations. And in my experience, I think they do see that there's a momentum to change it. And there's two reasons. One is, as I said, the external pressure, so they know it's going to happen. We already see actually problems in reimbursement for some of our medicine, as well as diagnostics, actually also IVD, which was also our business to have the better qualified IVDs, and we got reimbursement. We also see that this is getting more challenging, let's say. So I do think there is a change, and there's also the thought, if we understand patients better and get diverse patients, we might also get another pool, but that is still clearer. So whereas before we targeted to a certain population in the first world, we are starting to think into going other countries, diverse backgrounds, etc. So I do think they might realize that this might open better efficacy, and then they might still not have less sales because we have more understanding of different patient populations in other areas. Unfortunately, all needs to move at the same time. So we need to understand the patient better, we need to have the technologies, we need to have cheaper prices, and then of course the sales also work. So it is a complex interplay.
And so sticking with kind of the revenue model side of things, this question's actually pretty good, and I think it's relevant to both because it covers both biomarker discovery and also diagnostics. And the question I have here is there profit to be made from biomarkers and diagnostics? If you haven't been paying attention, I think Roche Diagnostics kind of gives you the idea that they've made quite a lot of money on diagnostics. But, you know, what is the current revenue model versus what is the ideal or preferred one? And I think, Sarah, as as you go from the biomarker discovery side, do you want to give a thought on that?
Yeah, I can give a thought on that. So I'm coming from the biomarker discovery side, but also from the startup side, right? So it's a little bit different there because we want to make revenues. We also have VC funding, so our investors have certain expectations of how the company should ideally perform. So we've actually tried a number of different business models in the history of the company. One of the first was more around really just discovering our own biomarkers as our own IP and then trying to sell those as assets to, say, diagnostic companies. This was challenging because we found that we couldn't actually take them far enough to get to a validated state where someone like Roche Diagnostics would be interested. And I can understand that from the perspective of a bigger company. But I think there's funding that's missing for that intermediate phase of, here's a discovery, I need now to have a clinical validation of it, but I need like 5 million at least to run that clinical study. Who's going to pay for that before I can potentially sell it? So that's one thing. But then if you do manage to get a product, then there's different ways that could go forward. So kind of an easy route, at least from the way I see it, is more like on the companion diagnostic side, because then it's obviously tied to sales of the drug itself, and or having something that's tied to a platform, which is kind of, I think, one of the main models that you use. Yeah.
So, and leading on, I've got another question that follows that quite nicely, and this is for Elke. Speaking of companion diagnostics, what do you see as the main obstacle of developing and commercializing companion diagnostics?
Well, I can just speak from internal. In our case, we are still separate units in a sense. So we have multiple business slots and areas and pockets. So it's really, I would even call this a little like inner politics kind of agreement. So you would need really super higher upper management endorsement and really seeing where the need for the overall company is. Otherwise, it's going to be a maneuvering through the sales part where everybody has, who's a current head of XYZ, they have their exact goals and they have their sales, etc. So it would need to be an agreement and both need to meet halfway. But this is for sure possible, I think, under the current setup even more than in the past, because we have by now the farm or personalized healthcare system being supported, and we have FMI being integrated into even Roche Diagnostics. And we do see that this is starting to happening even on the upper management, that there comes in general an endorsement, and then when it comes to the concrete thing, it's going to be still complicated.
So I'm just going to make one very quick clarification, because pharma are wonderful about our acronyms and we forget this.
Yes.
As we're on Novartis campus,
Yes.
we don't want to cause great panic on FMI being integrated into Roche Diagnostics.
Yes, the Roche FMI, sorry. Yes.
So that's the foundation medicine.
Yes, foundation medicine.
As opposed to Friedrich Miescher. So
yes.
We're not breaking any wild, crazy news
thank
today.
you. No. Yes, thank you. Yes. True. Yes.
Good stuff. So
Yeah, that would have been bad.
yeah, a little bit, yeah. So I'm just flicking through because I have a couple more questions on this one. No, I think we're going to move on actually and focus on what are the other questions that have been really interesting. And I think, Sarah, I'm going to flick back to you first of all in this one, because we've got a number of questions on regulatory pathway. So, where do you think at the moment, where do you think regulators are positioned with regards to trust for AI black box discovery of biomarkers, first of all?
It's a tricky landscape for sure. It's not straightforward. I think there's different routes that one can try to take. So one is more the translational approach that I mentioned. That's kind of what we've been doing at Skylight until now, is that we have kind of a separation between the real discovery, which in our case involves AI, and then the product or the biomarker, which is a translation of that discovery into some prototype or assay that is not relying anymore on an AI model. It's coming out of that AI model, but it's it's something that can be implemented on a standard piece of equipment and that, you know, can be done with a standard simple analysis. And then the other part of that we also try to bring is the explainability. So not having it be a black box, but having algorithms that allow you to open them up and understand what has been learned and why it has made the predictions that it's made. And that's something that we put a lot of emphasis on. And there are different ways to do that. You can take their sort of classical approaches developed in machine learning to do what's called like feature importance attribution, where you can ask, of everything that went into my model, what are the most important features for the prediction that it made? So that's one way that you could do it. Now with, you know, big LLMs like ChatGPT and Gemini and these things, I think in some ways they're even more of a black box, but you have this new sort of chain of thought reasoning that's starting to become more widespread, where you can actually ask the model itself to explain to you how it got to a certain conclusion. I still wouldn't necessarily trust everything. I would definitely take it with a grain of salt. But I think, yeah, from a regulatory perspective, one has to be careful. And I think also you can be strategic about how you position your product. So if you're building a medical device, there are all these different levels of classifications of medical devices, at least in the EU. And so, for example, I think I may say the wrong thing here, but I think it's class 2 is not directly making a diagnosis or making sort of recommending a treatment, but rather something like a clinical decision support system that would aggregate data, present it to a clinician, and maybe give some ideas or kind of contextualize things, but not necessarily making the final step. It's up to the humans still to make the final step. So that can be an easier regulatory route to go.
And what do you, from your side, Elke, how much interaction on the validation of new biomarkers do you have, and is this quite a large chunk of your no, not?
Yeah, no. I mean, I do see what's happening, because the ways of communication are short, but we are not covering it as much. We are really on the super early space. So I do know, because I work with the pharma teams, however, the hesitation to go into novel markers because of the regulatory, let's say, hurdles, and it's even, let's be honest, a combination of markers, and we are talking classical markers, that is causing sometimes problems for registering novel markers for certain indications, for certain treatments, than the classical ones that we might know. So, but we are still talking protein markers. So that's why I do think there's also the hesitation on the pharma side, is my current understanding, to move into more explore, even if they know certain markers might be better. So yeah, that's my understanding.
So I've got two questions that I'm going to go with here that have come from the audience that are they're big questions. So, you know, these are quite high level ones. So I think they're more on your opinions and your thoughts on this one. So we'll start with this one. What is your take on using quantum computing for precision medicine? There you go.
Okay.
Who'd like to start? Sarah, you're closest to me, so you're my first victim.
All right. Honestly, I don't think I'm going to have a lot of smart things to say about quantum computing, because it's not my field of experience. I think it's still pretty new, and I don't think there's much that can be really directly implemented now. I mean, if we talk about all the challenges we've talked about with kind of building up from combinations of proteins to omics to AI on standard computing and now quantum computing, I think that's even more farther in the future. But yeah, watch this space, I guess.
Yeah, very similar opinion. I mean, science-wise, it's very interesting, but what this can open, but to me, this is where currently reality stands for me. I maybe if I'm lucky, I see this when I'm in pension, that this is being used. I don't know. I'm just throwing something in the air, but yeah.
And along that lines, not quite so quantum computing heavy, but if you think about your role and what you do right now, day to day, how do you see AI changing what you do currently in the short to medium term? So in the next 2 to 3 years, what changes do you see coming up? Sarah, if you go on ahead.
I would divide it into two broad areas. So one is really the science, and then the other one is kind of accelerating the day-to-day, which I think applies to most of us probably. So, no, but honestly, it does change things. On the science side, I think, this is a field where there are a lot of really smart people working on this in academia. And people like Avi Vergev, Fabian Theiss, I could drop lots of names, but I'm sure there are going to be many more advances in terms of using AI to model deep biology, to model cells, and to try and grasp that complexity. Because actually, if you think about a human organism in terms of the number of proteins, the number of different molecules multiplied by the number of cells in a human, this is actually an order of magnitude more complex than the number of parameters that the most advanced natural language chatbots are trained on right now. So this is really, really complicated, but the technology is coming. So I do think that it's going to be really fascinating to watch where this goes in the next decade in terms of the biology. And then, of course, there's kind of the everyday work, but that it's also having a big impact in our space, but in plenty of different fields where you can start to deploy AI agents to speed up tasks like grant writing, like marketing and sales and all of these different things. So I think this will change. Certainly for startups, the way that we operate and the way that we think about our budgets and our personnel and this kind of things, it is changing already. I can tell you that. Yeah.
Do you think, just before I ask Elke on that one, because I really like the topic you touched on there, do you think that AI is being deployed differently in startups to large established companies? Do you think it has a bigger impact on startups?
Well, I can't say exactly what it's like in large established companies, but I think I think startups are well positioned to deploy it in a way that is impactful because we can be fast and agile, and while we absolutely care about things like data security and privacy, we can maybe afford to take a little bit more risks and try things out, and so I think in that way, it's just kind of part of our DNA to test things, to be fast, and if it doesn't work, we leave it behind. It's not that big of a deal. We don't have a huge multinational organization that we have to roll out coordinated changes across every computer. We're a team of 15 people, so we can move fast.
Yeah, that's definitely a plus.
Yeah. So Elke, from your perspective, if you think of your job at the moment, 2 to 3 years, what do you see AI will have changed?
Well, our team is still kind of new, so we, from the beginning on, I think, try to be resourceful, because I do think the biggest impact it will have for big companies is, they will also save, we could call it saving resources, right? Which is unfortunate. This might cost headcount, etc., people's jobs, but this is in the end what is happening. So the job markets are changing. So certain jobs you might need less of, whereas other jobs might still exist. And one big branch is we do, just to give an example, we have, as you might know, these IHC platforms to stain for tissue cancer, the previous acquired Ventana a long time ago. I mean, as you know, this is really AI driven for actually also the diagnosis. So we integrate AI systems also for the earlier readout, which then replaces jobs. Internally, same thing, regulatory things we use, we start to think about using AI. This is standardized. You could easily feed in certain requirements, and instead of having 5 medicals looking over it for half a year or whatever, I'm just making numbers up, but you can, of course, get certain things done by AI. For me, I personally love it because it saves me time. So I use the internal platforms for AI to, even sometimes check my emails cross, put PowerPoints in, anything, etc. And already this did cost my team less work to support me, even in parts, because I get it so fast done myself by just feeding it into certain platforms. It's amazing. So I'm quite positive, but I can see this usual thing, whatever you put in, it also comes out, right? So what AI, let's say, garbage in, garbage out. So if I do a really weird email, there comes still a weird email out.
Okay, so I think actually we're going to wrap up on that. We've got one last question, and this is from myself, and it's more of a future thinking area. Is first of all, thank you both for your engagement in the conversation. Thank you for the audience as well for the questions that you've submitted. But if you both had a think and you could make one suggestion on what could be changed to improve discovery and applications in biomarker in precision medicine, what would it be? Who wants to go first?
Well, I think in the end, it's something that's already been talked about. But for me, it would come down to having better aligned incentives, actually, across payers, reimbursement, but also regulatory, and aligning that with patient needs. Because I feel that we're often navigating this landscape where we're being pushed and pulled in different directions. One person wants this, one wants this, and the classic, as you said, pharma says, no, I don't want to shrink my pool of possible patients that I can treat. So, I think having incentives that would push the industry together in the same direction would go a long way.
It's a similar direction. I feel it counts to many places or many situations in our life. You do have to take care of business, but what is it you can maybe additionally to that doesn't harm you? Mm-hmm. And this is something, and it's the same when you ask for your help, you're on the street and you ask someone for a way or someone to give. We talked about this at lunch actually, so how much does it cost you to help someone else? So, and the same accounts for me for precision medicine. If you have to do your everyday business, that's fine. If you have to, but maybe you can do something little on top to make the field moving. I think with that mindset and incentive, then innovation moves, and then we can keep this forward.
Fantastic, and I think we'll leave it on that, folks. So in your day-to-day life, I think that's actually a really nice message for everybody. What else can you do a little bit on top of that will mean something to somebody else? And I think we'll finish on that. So thank you very much, everybody. Did he leave? No, he's there.
You can leave if you want.
Excellent. Thank you very much, Quiva, Elke, Sarah, for this insightful discussion. We announced it, and I think we did get this insightful discussion indeed. I would like to address a few thank you notes. Again, also Novartis, of course, for sponsoring this event series. We're super grateful that we can continue doing this at the Pavilion 4 times this year. I mentioned that I represent the Switzerland Innovation Park Basel area. We have work desks and we have offices free for health tech ventures. If you want to be among cool startups such as Scalite, Sarah is with us and the Day One team as well. So get in touch with me. And then I think there's some news on Day One, which I hand over to Ursula.
Yes, so we have you often here, and for those of you that follow us in social media or know about Day One for long, you might have seen that something exciting is coming next Monday. And we know we always have very brilliant minds here, both on site and online. So we wanted to give you the premiere. So you're the first ones to get to know and to get access. So you will see the pages not yet live, but next Monday. And I want to extend a big thank you to Quiva that you have seen and to Cecile here in the front. We are relaunching our accelerator and we want to welcome you to there. So if you know a startup, a health tech startup, so we don't do molecules, we stay as they want, we stay to our core. But if you know health tech startups that work on pharma R&D, especially on clinical development, please feel free to share the link. If you work in pharma or in any other sector and you think a really niche, tailored, and dedicated accelerator for those companies makes sense to you, feel free to reach out. I'll be here during the apero. And I really want to get Cecile because she's the person that is going to be coordinating it. Quiva, you have seen her for an hour and a half now, but she is the mastermind and the other person without whom it wouldn't be possible. So please go there and enjoy. You get a week ahead of everyone else. So thank you very much for joining, and hope you become part and you get to also enjoy the new journey that we take on day one. And with that, just save the date in your calendar. So next open mic is coming up very soon, May 26th. And we can already tell you it's going to be on the topic of tech for sustainability in pharma. And we're already lining up speakers, and it's going to be another amazing opportunity to be here together at the pavilion. So save the date and see you very soon.